Welcome back to the deep dive. Today, we aren't just looking at a bug. We are analyzing a biological tank. We are dismantling the genus Mycobacterium.
And tank is absolutely the right word. You know, in the world of microbiology, you've got your speed demons like E. coli. They just rely on replicating as fast as possible to survive.
Hit hard and fast.
Exactly. And then you have Mycobacterium. It doesn't care about speed at all. It's all about persistence.
Yeah.
It has built this fortress around itself and just decided to wait out any siege.
And we have a heady stack of sources today. Murray's Medical Microbiology, Chapter 22, is going to be our main guide for this. And we'll be pulling in some clinical pearls from Jeb's Chapter 23 as well.
Right.
Our mission here is really to move past just memorizing lists. I mean, everyone knows TB causes cavities and night sweats. We want to get to the mechanisms that let an organism survive inside the very cell that's designed to kill it.
That's the key. If you can visualize the architecture of this bacteria, specifically that cell wall, you don't need to rote memorize the drug list or the pathology at all. It flows from that.
The transmission, the immune response.
The diagnostic delay, everything. It all comes back to that wax.
Okay, so let's start right there, the waxy fortress. Murray describes the genus Mycobacterium as, aerobic, non-motile rods. But the defining feature, the thing that makes them a nightmare for the lab and for the patient, is that they just don't play by the rules of the gram stain.
Right. And this is a classic trap for medical students. Genetically, if you look at their lineage, they are gram-positive bacteria. They have a cell membrane. And then outside of that, they've got a thick layer of peptidoglycan.
So they should stain purple.
They should. But they've basically added this massive granulovation on top of that standard gram-positive house. So attached to the peptidoglycan is a complex layer of arabinogalactan,
which is a polysaccharide polymer.
Correct. And then anchored into that are the mycolic acids. And this is the star of the show. These are not your standard phospholipid fatty acids that are, 16 or 18 carbons long?
No, these are behemoths.
They're massive. We are talking 70 to 90 carbons in length.
That's an enormous molecule.
It's huge. And because they're so long and saturated, they form a wax. It's essentially a layer of hydrophobic wax surrounding the entire cell.
So when you pour the crystal violet dye on the slide for a gram stain, what's happening? Or rather, what's not happening?
It's like pouring water on a raincoat. It just beads up and rolls right off. It can't penetrate that wax. So when you look at the slide under the microscope, you see, well, nothing.
Ghost cells.
Exactly. You see these clear, unstained silhouettes where the bacteria are, and that's your first clue. But, clinically, we need to actually see them. So that's where the acid-fast stain comes in.
And it's worth explaining the physics of this because it really explains the name.
It does. Acid-fast always sounds like they move quickly or something.
Yeah, it's a bit misleading.
It's the opposite. It refers to their fastness, as in their ability to hold on. So to stain them, we use a lipid-soluble dye called carbolfutsin, but just pouring it on isn't enough. We have to literally force it past that mycolic acid shield.
And that's usually done with heat, right? The Zeal-Nielsen method.
Right. You're essentially boiling the dye into the wax. The heat gives the dye molecules enough kinetic energy to slip through that waxy lipid layer. And then, once the slide cools, the wax solidifies again, trapping the dye inside.
It's locked in.
It's locked in tight. Then comes the test. You wash the slide with a really potent mix of acid and alcohol. If this were Staph aureus or E. coli, that acid would strip the dye out instantly.
But the mycobacteria.
The wax protects the dye. They hold fast against the acid, hence acid fast.
And that's where we get those classic bright red rods against a blue background. But Murray points out that this wall, it's not just a staining annoyance. It comes with a really heavy metabolic price tag.
Oh, absolutely. Biology is always a trade-off. I mean, building a fortress out of 90 carbon fatty acids requires a huge amount of energy and raw materials. And it also really limits nutrient uptake because the wall is so impermeable.
Which directly explains the growth rate.
It's painfully slow. E. coli doubles every 20 minutes. Mycobacterium, tuberculosis, we're talking every 18 to 24 hours.
That is a massive difference.
Then it changes clinical practice entirely. You can't just order a stat culture for TB. You're waiting 4 to 6 weeks for visible colonies on a solid media like Lowenstein-Jensen.
Which is why we've had to develop other methods like PCR and the rapid liquid cultures, which we'll definitely get to. But there's another big implication of this wall: antibiotics. If nutrients can't get in easily.
Then drugs can't get in either. Correct. Most of our common antibiotics, like penicillins or cephalosporins, work by attacking peptidoglycan synthesis, but they can't even get to the peptidoglycan in mycobacteria. They just bounce right off that mycolic acid shield.
So this organism is naturally resistant to the vast majority of our antibiotic arsenal before it even develops a single mutation.
Exactly. You need very specialized drugs that are either lipophilic enough to get through or have specific transport mechanisms to breach that wall.
Okay, so before we dive into the specific diseases, let's just briefly organize the family tree, because Mycobacterium isn't just one bug.
Right. And we usually categorize them by growth speed and clinical syndrome. At the very top, you have the tuberculosis complex.
The slow-growing killers.
Yes. And then you have the non-tuberculous mycobacteria, or NTMs. And Jawetz mentions a classification system that, well, it's a bit old school, but it's still incredibly useful for organizing this in your head. The Runyon classification.
This divides them based on two things. How fast they grow and whether they make pigment.
Exactly. So group I are the photochromogens, like M. consasi or M. marinum. They produce this yellow pigment, but only when they're exposed to light.
Like a bacterial suntan.
And bacterial suntan, yes.
Yeah.
Perfect analogy. Then group 2 are the scotochromogens, like M. gordonei. They make pigment whether it's dark or light, they don't care.
And M. gordonei is the tap water one, right?
Yeah, that's the one. It's often called the tap water scotochromogen because it's in the plumbing. It rarely causes disease, but it terrifies new lab techs because it contaminates samples and looks like a pathogen until you properly identify it.
Then you have group 3, the non-chromogens.
No pigment. And this group is home to a real heavy hitter, MAC, the Mycobacterium avium complex.
And finally, group 3, the rapid growers.
And rapid is definitely relative here. We mean they form colonies in less than 7 days, which is fast compared to the month you wait for TB. This includes bugs like M. fortuitum, M. chelonae, and M. abscessus.
Okay, so the framework is established. Let's talk about the big one, the king, Mycobacterium tuberculosis.
A white plague.
Let's move to the battlefield. Pathogenesis. Murray describes this as a stalemate, but it has to start with an invasion.
And it starts with the breath. The vector is the aerosol droplet nucleus. These are tiny, less than 5 microns. They have to be that small to navigate the aerodynamics of the respiratory tract, get past the mucociliary escalator, and land deep down in the alveolar spaces.
Where the alveolar macrophage is waiting. Now, normally, a macrophage sees a bacteria, eats it, and destroys it. It's a routine patrol.
Right. The macrophage recognizes the bacteria using its surface receptors, specifically mannose receptors and complement receptor 3, or CR3. It engulfs the bacteria into a vesicle called a phagosome.
And the standard operating procedure from there is acidify the phagosome, fuse it with a lysosome, which is full of digestive enzymes, and just obliterate the invader.
That's the plan. But M. tuberculosis has a pre-programmed countermove. It actively inhibits that maturation process, and we actually know the molecular mechanism. It involves a protein called early endosomal autoantigen 1, or EEA1.
Okay, unpack that for us.
So for a phagosome to fuse with the lysosome, its membrane needs this specific bridging molecule, EEA1. TB essentially interferes with the macrophage's ability to get EEA1 onto the phagosome membrane.
So the docking port is broken.
Effectively, but it's even deeper than that. The bacteria also block the accumulation of the VATPase. That's the proton pump that drives acid into the vesicle.
So the phagosome never gets acidic.
It stays at a pH of about 6.4, which is really mild. A normal phagosome drops to a pH of like 4.5. So the bacteria creates this comfortable, neutral pH safe house inside the very cell that's meant to kill it.
It's not just hiding, it's hijacking the real estate.
And it's replicating. And this is where cord factor comes in. This is a term that shows up on every single board exam, but the biology behind it is just fascinating.
Trehalase 6,7,6-dimyclit.
That's the chemical name. It's a glycolipid in the cell wall. When virulent strains of TB grow, this molecule causes them to stick together end to end, and they form these long, serpentine, rope-like structures under the microscope.
So if you see that cording, you're looking at a killer strain.
Exactly. The virulent strains don't do this. The cord factor isn't just for looks. It's actually toxic to the mitochondrial membranes of the macrophage. It disrupts the host cell's energy production, and, crucially, it prevents the migration of neutrophils. It lets the bacteria set up the granuloma on its own terms.
Okay, so the macrophage has been compromised. The first responder is captured. Move 3 must belong to the host defense.
Right. The infected macrophage realizes it's in deep trouble. It starts secreting interleukin-12 or IL-12 and TNF-alpha. This is a distress flare.
And the intended recipient of that signal is the T helper cell.
Specifically, the Th1 subset. This is so vital. The antibody response, the Th2 response, is completely useless here because the bacteria are all hiding inside cells. Antibodies can't reach them.
You need cell-mediated immunity.
You need it. The IL-12 calls in the T cells, the CD4 positive cells.
So the T cells arrive and they secrete interferon gamma, IFN gamma.
This is probably the most critical cytokine in the entire TB story. IFN gamma is like the super soldier serum for the macrophage. It binds to the macrophage and just supercharges it. It forces the production of nitric oxide synthase,
which generates reactive nitrogen intermediates,
which are finally toxic enough to kill the bacteria. And IFN gamma also overcomes that blockade we talked about earlier. It forces the phagosome to fuse with the lysosome.
So the T cell basically yells at the macrophage, stop messing around, acidify that vesicle, and digest it now.
That's a perfect way to put it. And this whole IL-12, IFN gamma loop is the linchpin of the whole defense. If a patient has a genetic mutation in their IFN gamma receptor, or if they have autoantibodies against IFN gamma, they get overwhelmed by mycobacteria that are normally harmless to us.
So, assuming the host is competent, the T cells arrive, the macrophages get activated, the battle intensifies. But even then, the body often can't kill all the bacteria.
No, it often can't. So we enter the stalemate phase, the granuloma.
The prison.
It is a prison. The body just walls off the infection. In the center, you have the infected macrophages. And the battle is so intense that the tissue dies, forming what we call caseous necrosis.
Cheesy necrosis.
Literally. It looks like soft cheese. And surrounding that necrotic center, you have these giant cells, which are just fused macrophages, and then a ring of T cells and fibroblasts, creating a fibrotic cap around the whole thing.
And inside that cheesy center, the bacteria aren't dead.
They're dormant. They're in a state of latency. They slew their metabolism down to a crawl. They can survive in that hypoxic, nutrient-poor environment for decades.
And that's latent TB. That's the state of about a quarter of the world's population, just walking around with a walled-off prison in their lungs.
Precisely. And as long as the warden, the immune system, stays awake and on guard, the prisoners stay put.
Which brings us perfectly to clinical disease and epidemiology. We have primary TB and reactivation TB.
So primary TB is that initial infection, and it usually occurs in the middle or lower lung fields.
Why there?
It's just airflow dynamics. That's where the particles tend to settle after you inhale them, and this forms what we call the GON complex.
We should define that clearly for everyone.
The GON complex is the combination of two things: the initial parenchymal lesion in the lung tissue itself, plus the enlarged hilar lymph node that drains that area. And usually this just heals, it calcifies, and you might see it on an X-ray 40 years later as a little white scar.
But then, years or even decades later, the immune system starts to wane. Maybe it's old age, maybe it's steroids, maybe it's chemotherapy. And we get reactivation TB.
The prison break. And interestingly, reactivation usually happens in a totally different location. The lung aposes, the very top of the lungs.
Why then move upstairs?
Oxygen. Tuberculosis is an obligate aerobe. It needs oxygen. And the top of the lungs has the highest oxygen tension, the highest PO2, because gravity reduces blood flow to the apex. That leaves the air sacs fully distended and rich with oxygen.
It's a 5-star resort for aerobic bacteria.
It's a spa, and this is where the real damage happens. The immune response goes into overdrive trying to catch the escaping bacteria. It liquefies the caseous necrosis, and that forms these large cavities.
And those cavities erode into the airways,
causing the coughs, and they erode into blood vessels.
causing hemoptysis, coughing up blood.
Exactly.
Okay, we absolutely have to talk about the perfect storm, the interaction between HIV and TB.
It is probably the most devastating synergy in all of infectious disease. Remember we said the granuloma, the prison wall, depends on CD4 T cells.
Right, they're the wardens.
Well, HIV targets and destroys CD4 T cells. So if a patient with latent TB gets HIV and their CD4 count starts to drop, the walls of that prison just dissolve.
And you get rapid, widespread dissemination.
Everywhere. The lungs, the liver, the spleen, the brain. And here is the really scary part for a clinician. Because the patient lacks the T cells to mount an inflammatory response, they might not form a cavity or an infiltrate.
So the chest X-ray can look normal.
It can look completely normal, no inflammation, no shadows, and yet the patient is teeming with bacteria. It's a diagnostic nightmare.
Speaking of diagnosis, let's get into the lab. We've talked about the acid-fast stain, which is our rapid screening tool, but now we need to talk immunodiagnosis. So The skin test versus the blood test.
The tuberculin skin test, the PPD, has been used for over a century. You inject a protein derivative under the skin, you wait 48 to 72 hours, and you're looking for induration, a hard bump.
And this is measuring type IV delayed hypersensitivity. Basically, are there memory T cells in the body that recognize these proteins?
Exactly. But there is a massive, massive flaw in the PPD, and that's specificity.
The BCG vaccine.
Right. The BCG vaccine is used in many countries, though not really in the U.S. It's derived from M. bovis, and it shares a lot of antigens with M. tuberculosis. So if you were vaccinated as a child, your T cells will react to the PPD, you get a false positive.
That's a huge problem in a globalized world where patients come from all over. So how do we fix it?
We use the IGRA, the interferon gamma release assay. The common brand name you'll hear is quantiferon gold. This is a blood test.
How is it different?
Well, it takes the patient's blood and stimulates their T cells with antigens that are specific to M. tuberculosis, specifically proteins like ESAT6 and CFP10. Crucially, these proteins are absent from the BCG vaccine strains.
So if you have a positive quantiferon, it's not the vaccine.
Correct. It's remarkably specific, a huge leap forward.
Okay, let's talk about the gold standard, culture. We mentioned it takes weeks. What are we growing it on?
The classic medium is Lowenstein-Jensen. It's an egg-based medium. It's got eggs, potato flour, glycerol. It looks like a green slant in a tube.
And what do the colonies look like?
The classic description is rough, buff, and tough. They're dry, they're crumbly, and they're beige. They kind of look like cauliflower.
But waiting 4 to 6 weeks is really hard when a patient is actively coughing and potentially spreading the disease.
Which is why not nucleic acid amplification testing, or PCR, is really the standard of care now. We can detect the bacterial DNA in a sputum sample in hours. And crucially, we can also look for resistance genes at the same time.
Which is a perfect segue to treatment, Section 5.
Yeah.
The strategy here is pretty unique because of that whole stalemate concept we talked about.
Right. You're treating a slow-growing, persistent organism that hides inside cells and necrotic tissue. You cannot treat TB with just one drug. You will select for resistance almost immediately.
So we use the RIPE regimen, RIPE.
Rifampin, isoniazid, which we call INH, pyrazinamide, and ethambutol.
Let's break down the mechanics of a couple of those, because the boards absolutely love them. Isoniazid.
So INH works by inhibiting the synthesis of mycolic acids. It stops the fortress wall from being built. But here is the mechanism you absolutely need to know. INH is a prodrug.
Meaning it's inactive when you swallow it.
Right. It needs a bacterial enzyme catalase peroxidase to activate it, and this enzyme is encoded by the KG gene.
So if the bacteria gets a mutation in that KG gene,
the drug never gets activated, it's useless, and that is the primary mechanism of resistance to INH.
And what about rifampin?
Rifampin inhibits the bacterial RNA polymerase. The gene involved there is RP-Phe. A mutation in RPB confers resistance.
So why do we need pyrazinamide? What's its special role?
Pyrazinamide is the sterilizing drug. It is particularly effective against those dormant bacteria that are hiding inside the acidic environment of the macrophage lysosomes. It's the drug that helps shorten therapy from 9 months down to 6.
So the standard course is 2 months of all 4 drugs, followed by 4 months of just INH and rifampin.
That's right, assuming the bug is susceptible. If it's not, we enter the very scary world of MDR and XDR-TB.
Multi-drug resistant TB.
That's defined as resistance to at least isoniazid and rifampin are 2 best drugs.
And then there's extensively drug-resistant TB, XDR-TB.
That's MDR plus resistance to a fluoroquinolone and at least one of the injectable agents. These cases are absolute nightmares to treat. We're talking 2 years of therapy, really toxic second-line drugs, and often surgical resection of the lung.
It's a good reminder that while we have antibiotics, that waxy fortress is constantly evolving its defenses.
Absolutely.
Okay, let's shift gears. We have covered the king. Let's look at the ancient, Mycobacterium leprae, Hansen disease.
This organism is just. It's biologically bizarre. It's an obligate intracellular parasite. In fact, it's undergone what we call reductive evolution. It has literally thrown away a huge chunk of its genome because it relies on the host cell for almost everything.
Which means we can't culture it in the lab.
Not on artificial media, no. You can't just put it on a Lowenstein-Jensen slant. To study it, scientists used to have to grow it in the footpads of mice.
And the armadillo?
The 9-banded armadillo, yes. It's a natural reservoir for the bacteria in the southern United States.
Why the armadillo? What's so special about it?
It's their body temperature. Armadillos have a core temp of about 32 to 33 degrees Celsius, and M. leprae's enzymes just happen to function best at cooler temperatures.
Which explains the pattern of human symptoms perfectly.
Exactly. Leprosy affects the cooler parts of the human body, the skin, the superficial nerves, the nose, the anterior chamber of the eye. It completely spares the warm internal organs.
Now, the clinical presentation is a spectrum, and this is maybe the best example in all of medicine of that Th1 versus Th2 immune balance.
Oh, it's the textbook definition. On one end of the spectrum, you have tuberculoid leprosy.
In tuberculoid leprosy, the patient has a very strong cell-mediated or Th1 response.
Right. Their T cells are active, they're making IFN gamma, they are containing the infection. So clinically, what you see is paucibacillary disease.
Meaning few bacteria.
Very few. You'll see these hypopigmented, dry skin patches. And they are anesthetic, they're numb, because that strong immune response actually damages the peripheral nerves in that area. And if you do a skin test, the leprae test, it's positive.
Because the T cells are there to react. Now let's flip the coin. Lepromatous leprosy.
Here, the immune system makes a fatal error. It mounts a Th2 response, an antibody response.
Which, as we already established, is useless against intracellular bugs.
Completely useless. The macrophages get packed with bacteria, literally millions of them. We call them foam cells or globi.
So this is multibacillar.
Very. The bacteria spread everywhere in the skin. You get these disfiguring nodular skin lesions, a thickening of the facial skin called lion facies, loss of the eyebrows, destruction of the nasal cartilage.
And the lepromin skin test in this case.
Negative. The body is awash in bacteria, but the specific T cells aren't there to mount a response. It's a state of anergy.
And treatment will depend on where on that spectrum you fall.
It does. Tuberculoid gets rifampin and dapsone. Lepromatous gets those two plus clofazimine. And again, you're treating for a long time, 1 to 2 years.
All right, let's pivot from the ancient to the opportunists. The Mycobacterium avium complex, or MAC.
This is a totally different beast. It's an environmental organism. It's in soil, water, dust. Unlike TB or leprosy, this is not transmitted person to person. You inhale it or ingest it from the world around you.
And Murray outlines 3 really distinct clinical presentations. Pattern recognition is everything here.
It is. First is the classic presentation, the smoker. This is usually a middle-aged man with underlying lung disease like COPD, and he gets cavitary lesions in the upper lobes that look for all the world like TB.
Okay. And the second one is the one with the fascinating name.
Lady Windermere syndrome, which is named after the Oscar Wilde play, Lady Windermere's fan.
And who fits this profile?
It's typically an elderly, non-smoking woman, and the theory is that she habitually suppresses her cough, maybe out of social politeness or just habit.
Why does that lead to this specific infection?
It prevents the clearance of secretions, but the anatomy here is crucial. The right middle lobe and the lingula, which is on the left, have bronchi that are long, narrow, and angled pretty sharply. They rely heavily on a vigorous cough for drainage.
So if you don't cough.
Secretions just pool in those specific lobes. MS sets up shop, you get bronchiectasis and infiltrates, but you usually don't see cavities.
And then there's the third presentation, the AIDS presentation.
Disseminated MS. This was ubiquitous before we had good antiretroviral therapy. When a patient's CD4 count drops below 50, MS can cross the gut wall and it just spreads everywhere.
The liver, spleen, bone marrow.
The bacterial load is just staggering. You can literally see the blood teeming with acid-fast bacteria. The patient wastes away with fever and diarrhea.
And the treatment for MSC involves macrolides.
Yes, clarithromycin or azithromycin is the backbone of the therapy, usually combined with ethambutol and erythromycin. And for AIDS patients with those very low CD4 counts, we used to use azithromycin as prophylaxis to prevent this.
Let's move on to the rapid growers, M. fortuitum, M. cheloni, M. abscessus.
These are the speedsters of the family. They grow in less than a week, and they're environmental bugs that tend to get in through trauma or foreign bodies.
There's a really specific high-yield scenario mentioned in Jowett's regarding M. fortuitum.
The nail salon outbreak.
Tell us about that.
It was a famous outbreak that was traced to whirlpool foot baths. The screens in the baths were just teeming with M. fortuitum. Customers would shave their legs, which creates these tiny microabrasions, and then they'd soak their feet.
And the bacteria would get into the hair follicles.
Exactly. And that resulted in furunculosis, these nasty, non-healing boils all over their legs. And it's resistant to our standard skin antibiotics. You need weeks of a specific therapy like doxycycline or a sulfonamide.
A couple more quick hits from this group, M. marinum.
That's the fish tank granuloma. If you're cleaning a fish tank and you have a cut on your hand, this bug can get in. It forms a granulomatous ulcer.
And it likes the cold, right?
Yes, it grows best at about 28 to 30 degrees. And interestingly, it can spread up the lymphatics, which can make it look like sporothrix, the rose gardener's disease.
And M. cansasi.
That's the photochromogen we mentioned earlier. It causes a lung disease that is indistinguishable from TB. But the good news is it's not contagious person to person. Treatment is usually rifampin, ethambutol, and INH.
All right, section 9. The imposter. Nocardia.
Nocardia is so tricky because it's not a mycobacterium, but it sits at the same table microbiologically. It's an aerobic, gram-positive rod.
But it looks like a fungus under the microscope.
It does. It branches. It forms these long, filamentous, hyphae-like structures. If you just glance at the gram stain, you might mistake it for a mold.
But the key exam differentiator is the acid-fast stain.
It is weakly acid-fast.
What does that mean physically? What's different?
Its mycolic acids are shorter. They're around 50 carbons long instead of 90. So if you use the standard harsh acid alcohol wash that we use for TB, you will wash the dye right out of nocardia.
So you have to be more gentle?
You have to use a weaker acid, like 1% sulfuric acid. And if you do that, nocardia holds on to the red dye. This is so critical for the lab. If you just order a standard AFB stain, you might miss it. You have to specifically ask for the modified acid-fast stain.
And nocardia is found in the soil, so who gets sick?
The immunocompromised. Patients with T-cell deficiencies, so think lymphoma, transplant recipients, people on high-dose steroids.
And there's a clinical triad to remember.
Yes. Number 1, pulmonary disease pneumonia with cavitation. It looks just like TB. Number 2, CNS. It has a remarkably high affinity for the brain, so it causes brain abscesses. And number 3, cutaneous. It can cause skin abscesses or a mycetoma.
So if you have a patient with pneumonia and a brain abscess,
you have to think nocardia.
And the treatment? There's a great mnemonic for this.
SNAP. Sulfonamides for nocardia. Actinomyces gets penicillin.
So high-dose trimethoprim sulfamethoxazole, or Bactrim, is the drug of choice.
Exactly.
And finally, just a quick nod to Rhodococcus.
Rhodococcus secreti. It's a gram-positive coccobacillus that is also weakly acid-fast.
What's the classic association?
Horses. It's a common pathogen of foals. So you want to ask about exposure to farm animals or manure. In immunocompromised humans, especially those with HIV, it can cause a really destructive cavitary pneumonia.
And the visual on the plate is pretty distinct.
It is. It forms these beautiful salmon pink colonies.
Okay, we have covered the fortress, the king, the ancient, and the imposters. Let's try to synthesize all of this.
The overarching theme here is persistence. These aren't speed demons. They are all about playing the long game. They build these waxy walls to exclude drugs and enzymes. They hijack our own immune cells to create safe houses for themselves.
And the outcome in almost every single one of these infections depends on the host's cellular immunity.
It all comes down to that CD4 count and that IL-12 and IFN gamma axis. If that axis holds, we contain them. If it breaks, the fortress expands and the disease wins.
It's a reminder that in the fight against Mycobacterium, the most important variable is very often the host, not just the bug.
Well said.
On that note, let's lock this knowledge in with a quick recall session.
Let's do it.
I'm going to ask a single best answer style question. I'll pause for a few seconds to let you think it over, and then our expert will give the answer and the context behind it. Ready?
Ready.
Question 1. A patient with HIV and a CD4 count of 25 presents with fever, weight loss, and diarrhea. Blood cultures eventually reveal acid-fast bacilli. Which prophylactic antibiotic is historically indicated when CD4 counts drop below 50 to prevent this specific infection?
Okay, a classic scenario. Think about the bug that causes disseminated disease at that CD4 count.
The answer is azithromycin or clarithromycin.
This is the classic presentation of disseminated MAC infection. Prophylaxis with a macrolide was standard when the CD4 count was less than 50, though guidelines do evolve with better antiretroviral therapy. You can review this in the Murray section on Mycobacterium avium complex.
Question 2. A microscopic exam of a sputum sample reveals serpentine cords of acid-fast bacilli. What specific glycolipid virulence factor is responsible for this morphology and for preventing neutrophil migration?
This is all about what makes virulent TB look and act virulent.
The answer is cord factor, or trachelose 6-expicit demyclate.
Cording is a key hallmark of virulent M. tuberculosis. Cord factor is toxic to mitochondria and is essential for the proper formation of a granuloma. Review the Murray section on M. tuberculosis pathogenesis.
Question 3. An elderly woman with no history of smoking presents with chronic cough and infiltrates in the lingula and the right middle lobe. She admits to habitually suppressing her cough. What is the most likely pathogen?
Think about the name and the specific anatomy involved.
The answer is Mycobacterium avium complex, MAC.
This is the classic Lady Windermere syndrome phenotype. The anatomy of the middle lobe and lingula predisposes them to poor drainage, especially if the cough reflex is suppressed, allowing MAC to take hold. Review the Murray section on MAs.
Question 4. A patient on chronic high-dose steroids presents with a lung nodule and a ring-enhancing brain lesion. An aspirate shows filamentous, branching, gram-positive rods that are weakly acid-fast. What is the treatment of choice?
Okay, put the pieces together. Immunocompromised host, lungs, brain, and that unique staining pattern.
The answer is trimethoprim sulfamethoxazole, TMP-SMX.
The organism is nocardia. The key features are the branching morphology, the weak acid fastness, and the tropism for the brain. Remember the mnemonic SNAP, sulfonamides for nocardia. You can find this in the Murray section on nocardia.
Question 5. A patient from a tropical region presents with hypopigmented anesthetic skin patches and a thickened ulnar nerve. A skin biopsy shows granulomas but very few acid-fast bacilli. Which arm of the immune system is dominant in this patient?
Think about the immune spectrum of leprosy. Lots of inflammation, but few bugs means what?
The answer is the Th1 cellular immune response.
This is tuberculate leprosy. The strong cellular response, driven by things like IL-2 and IFN-gamma, successfully contains the bacteria, leading to possibacillary disease but also causing nerve damage. Review the Murray section on M. leprae.
Question 6. Which scanning method for Mycobacterium tuberculosis is most appropriate for rapid screening of clinical specimens, mainly due to its high sensitivity and the ability to read it at a lower magnification?
So not the classic confirmatory stain, but the best one for screening a lot of slides quickly.
The answer is the truant fluorochrome stain, which uses auramine-rhodamine.
While Ziehl-Neelsen is the classic confirmatory stain, the fluorochrome stain causes the bacteria to glow brightly against a dark background, making them much easier and faster to spot during screening. Review the Murray section on diagnosis.
And the last one, question 7. Resistance to isoniazid, or INH, is frequently mediated by a deletion or mutation in which specific bacterial gene that codes for a catalase peroxidase.
Remember, INH is a prodrug. What does it need to get switched on?
The answer is the CATG gene.
INH requires the bacterial catalase peroxidase enzyme, which is encoded by the CATG gene, to become active. If the bacteria loses CATG function, the drug is never turned on, leading to high-level resistance. You can review this in the Jopwit section on drug resistance.
Excellent work. If you manage to get those, you are in a very good place for your exams.
And just remember, don't just memorize the lists. Visualize that waxy wall. It really does explain almost everything.
Thanks for diving deep with us. We'll see you next time.
Stay curious.